Hello everyone, welcome to Global Compliance Panel live webinar on HACCP validation and verification. My name is Ron and I'm going to be your host for today's session. On behalf of the Global Compliance Panel team, I'd like to thank you all for being part of this event. Today's webinar will be presented by Melinda Allen. A few words about Melinda before we could start off today's session. Melinda Allen is a food safety and quality consultant in the food industry. Melinda has had a long and dedicated career of quality assurance and food safety leadership with companies such as Yum Brands and Quiznos. Her consulting clients have included leaders in the field such as Burger King, Panda Express, and Popeye's Louisiana Kitchen. She continues to work with many of these and additional clients. Melinda and her team of experienced professionals are available for quality assurance and food service auditing, specification and program development and training, product commercialization. We are honored to have Melinda with us to present today's webinar. Now, ladies and gentlemen, before we begin, I would like to inform you of the program outline for this training session. This webinar is for 16 minutes duration. First, Melinda will take you through today's webinar, highlighting the areas that will be covered, and she will then share with you her presentation. And I would also like to inform all our attendees, once a part of this teleconference, your phone lines will be placed on mute and would remain so until the Q&A window begins towards the end of the webinar. And I also request all to hold back your questions until the Q&A window begins. 10 minutes of your time will be allotted for the Q&A, during which your questions will be answered. Now, if for any reason you get logged out of this training session or teleconference, please follow the same procedure to join in again. And now that we're all ready to start, I request Melinda to take it from here. Melinda?
Thank you so much, Ron, for that introduction, and I thank everyone for joining us today. Hope to have a very productive session. Today we're here to talk about HACCP validation and verification. It's a critical part of the program that is also being emphasized by inspectors with respect to FSMA coming in inspection cycle. So I wanted to make sure that you all are very prepared to be audited against this section of the regulation, as well as its pertinence to any other types of certification or standards that you're being audited to. So without further ado, I'd like to go through what I call the course outline or the agenda. So for this session, we will talk about the definitions of both validation and verification that differentiate the two. And then we'll talk about how this element of HACCP is important to GFSI compliance. From there, we'll get directly into how you verify and validate your HACCP program, specific types of that validation being scientific and technical. We'll talk about the documentation, reassessments in terms of how you will go about revising the program as needed, keeping it up to date and scientifically relevant. And finally, you'll have an appendix of some helpful documents like a questionnaire quiz, as well as reference materials and a login sheet for documenting your training. So then the next slide. Slide 3 is the definition of validation. So it's important to know the difference between validation and verification. Validation is the process of demonstrating that your HACCP system, if operated as designed, can adequately control the identified hazards to produce a safe product. And what that means is that in this respect, you are looking to make sure that the program has been set up correctly, that the testing or the monitoring or whatever actions that you're completing will actually achieve the desired result. It can also refer to the scientific or technical justification or documented basis of the system. So why you think that the actions that you have implemented should work. That we should always do beforehand, so looking through scientific literature, working with colleagues in the industry, looking through industry documents, and we'll get into this, we'll reiterate this a number of times because these are the important ways that we validate any of our scientific hypotheses. And then finally, the initial practical demonstration of proving the system will perform as expected. So gathering data, testing out our hypotheses, testing out our program, so to speak, and figuring out if we can, in fact, provide evidence that what we have designed is, again, effective. So on the next slide, number 4, we have the definition of verification. So the first step is always to validate the program, to validate what our hypotheses are, to wrap up that design phase. Verification is more of an ongoing activity. So it's the use of methods, procedures, or tests, in addition to those used for monitoring, to determine if the HACCP system is in compliance with the plan, or whether the plan needs modification or revalidation. So as we're verifying the program, this is our daily activities of reviewing documentation, reviewing test results, reviewing our reports to see that our program is working as a program, not that our scientific hypothesis is working. So let me give you an example. If you have, say, for instance, a kill step, which involves heat treatment of a protein or a product ingredient design finished product, the validation would be, what is the temperature required to get killed? The verification would be, have I achieved that temperature throughout my product, throughout the day, for every single piece of item that's gone through? What makes this confusing is there is an element of verification and validation. So as you're doing your scientific study, you want to make sure that you have statistically valid or at least enough data points to ensure that your solution or your activity is effective across your entire system. If you have an oven bed which is, 4 feet wide and is progressing at a determined speed, you want to make sure that every piece of or every item that comes through that bed or every inch of product is achieving the temperature. So that would be verification. Again, validation would be, will that temperature, will those speeds, will that process consistently deliver what you need it to deliver? Just to clarify further, when you're dealing with regulatory agencies, they will come out and provide a verification of your program. So you have your agency verification, your plant verification. This is slide 5. FDA, USDA have their inspectors or agents that when they're reviewing your program, they're looking either at verification or validation, and we will clarify what they're looking for in each different respect. You have your plant verification, and again, that's what you do when you are daily reviewing and weekly reviewing documentation, signing off at a supervisory or managerial level that the employees who are specifically responsible for monitoring HACCP and prerequisite programs are, in fact, meeting all the objectives from the procedures. So So they are achieving the temperatures or checking the pH or achieving the clean food surface contact. So whatever that critical control point is, we want to verify that they're in fact meeting that limit. The next slide is number 6, agency verification. Again, I say not to be confused with the plant verification as we just discussed. The Food Safety Inspection Service, or FSIS, is verifying that the scientific and technical materials exist and provide a scientific and technical basis for the CCPs and critical limits. So they're doing what I would call a document validation. Validation through documentation. They're just making sure that your hypothesis is scientifically sound, that you have used some sort of basis that is industry and expert accredited for setting up your critical control limits and designing your process. For instance, you would never think that you were going to achieve the water activity level for a dry product without some sort of process that's going to dry the product. They're looking to make sure that we're not sitting in a room and just making up processes and limits off the top of our heads, or using anecdotal or what do you call that, mom and pop kind of knowledge. It has to have scientific basis based on studies, literature, white papers, published papers, industry regarded and accepted, regulated knowledge. So that's the agency verification. So the agency on slide 7, when they're conducting their verification, they've identified 4 processes or steps in the establishment verification of the HACCP system. 1, scientific and technical processes to verify that all critical limits at CCPs are adequate and sufficient to control hazards that are likely to occur. And again, that's also known from our perspective as validating the process. So they're looking at your documentation, calling it verification. The process of looking at your validation documentation is called verification from the agency's perspective. Then secondly, the assurance that the HACCP plan functions properly through frequent review of the plan, verification, again, checking records, review of records, and determination that appropriate decisions and product dispositions occur when deviations occur. So again, what we're walking through is what they do, what the agency does when they come out to review or verify your HACCP plan. So they're not conducting any in-house studies. They're not going back and doing any testing, taking samples. At this point, they're calling it verification because all they're doing is reviewing, again, your documentation, same as you're doing. Number 3, documentation through periodic review to ensure the accuracy of the HACCP plan, including an on-site review and verification of all flow diagrams, critical control points, critical limits, monitoring procedures, corrective actions, and records. So this is where they're just, again, doing a document review of your HACCP plan. And finally, regulatory verification that the plan is functioning satisfactorily through overall process validation, including any or all of the verification steps listed above, plus final product testing to demonstrate compliance with regulatory as well as other desired performance levels. So again, they may decide to pull some of your product and do a test to ensure that it meets the desired limits. We're talking a sampling that is much, much less than what you would need to do for validation. They're not collecting necessarily any statistically significant or significant number of samples. They're just going to pull a limited number of products to randomly check to see that you are in fact meeting the limits that you set forth. So all of this is what the FDA, USDA, FSIS will do if they say they're going to come in and verify your HACCP program. This is in no respect a substitution for your third-party review and your internal validation of your own HACCP program. So on slide 9, we want to get into what is it that we do to validate the HACCP program. So starting out, we are food scientists. This is a science. HACCP is a science-based, started with NASA, very data-intensive program. So where do we want to start is with scientific or technical basis for our decisions. So we're going to look for reference documentation, anything that we can find from peer-reviewed scientific journals, challenge studies, published studies, even our own in-house data. The most important word there is being, data and studies, so that again, we are using scientific literature to establish whatever control limits that we're placing on our micro hazards, our physical hazards, our chemical hazards. Chemical exposure levels need to be based on maybe their EPA standards, maybe their GRAS standards, but some number that has been scientifically developed in the industry that tells us what's a safe limit. Physical hazards. What is the size that would cause injury? Again, HACCP is an injury-based program. So what is the size of this item that we're trying to preclude or eliminate from our product that would cause injury to the user? And finally, micro limits. What are the accepted micro limits that we can have present in the product and still make a safe product? All of these, again, need to have a scientific or technical basis. So moving from there into the next slide, we want to talk about why is this so important? Why are we, now that we know what validation and verification are, what's the difference and what's the basis? Why is this something that has become so important in our industry? Two reasons. The first we've already gone through, which is agency verification, that with the passage of new food safety regulation, this has become a mandatory practice for all manufacturers, and more companies are being required by their customers to be in GFSI compliance. And what is this? This would be our food safety international standards that are utilizing several different standards or schemes or audit protocols to assess our ability to make safe foods. We will just go through a number of those standards and what they specifically ask for, reiterating why it's important for us to have a validated and verified HACCP program. So prior to the implementation of control measures to be included in operational PRPs and the HACCP plan after any change therein, the 8.5.2, the organization shall validate, the 3.15, that the selected control measures are capable of achieving the intended control of the food safety hazard for which they are designed. And the control measures are effective and capable of, in combination, ensuring control of the identified food safety hazard to obtain end products that meet the defined acceptable levels. That's FSIS 22000, which is a growing international standard used by many of the global multinational companies. And it's section 8.2 under their verification and internal audit section. So all of that that I just read really just reiterates what we talked about in terms of the definition of validation. You could substitute that in for that definition. The next standard is SQF, which many of you are probably even more familiar with because it has been probably the #1 implemented scheme in the United States. But in section 2.14 of that standard, the management review section, item 292 says the HACCP food safety team shall validate each CCP. Documented evidence shall show that the control measures selected and critical limits identified are capable of consistently controlling the hazard to the specified acceptable level. And then in 2.14.1, appropriate changes resulting from the review shall be incorporated into the HACCP plan and or prerequisite program, fully documented, and validation recorded. So this just again reiterates that verification and validation are not only critical to regulatory agencies, but also to our third-party standards that are being required of customers. In slide 12 is the BRC standard, wherein clause 2.7.3 states that the HACCP food safety team shall consider the control measures necessary to prevent or eliminate a food safety hazard or reduce it to an acceptable level. Where the control is achieved through existing prerequisite programs, this shall be stated and the adequacy of the program to control the hazard validated. Consideration may be given to using more than one control measure. So again, each of the standards that we are being audited against is specifically requiring that this validation take place. So let's move to the next slide, and the next topic is going to be the scientific or technical basis, how we go about exactly establishing that. We have established that there is an agency need to have these programs in place. There is a audit need to have this element of the program in place. So let's talk about nitty-gritty, how do we get this done? So we've got the 3 types of hazards: physical, biological, and chemical. On the next slide, we've got examples of what those can be. Important part being specificity. So after we've identified physical, chemical, and biological hazards, we have to specifically, very clearly define exactly what we're controlling. Is it the size of a physical hazard? Is it the colony count of a microbiological hazard? Or is it the concentration of a chemical hazard? Slide 15 just reiterates that. And on to slide 16, where we talk about the actual way we identify these items. So the documentation must identify all associated factors or conditions. So if we are going through our process, we want to look at each step and see when we might be introducing a particular hazard. This is related to validation in that as we're doing our validation study, we've identified the hazard and we're checking or testing it at the step in which we expect to eliminate it. Now, the scientific data that we're using in order to explain why we think our hazard is being controlled has to be very specific as well. So it needs to identify the specific hazard or pathogen, and this is on slide 17. So we're identifying the specific control parameters, meaning upper and lower limits, meaning process controls such as speed, temperature, whatever we're using in terms of setup for this process, and whatever end result we expect to achieve, like an upper or lower limit. Those things need to be well documented in either the data that we're using as justification or reference material, or the report that we're generating to show that we have in fact created our own data. So on slide 18, we talk about the documentation because obviously all of this needs to be documented. So we've got our supporting document that either we are gathering, so we've got a study which has been published. We have a practical demonstration where the data has been collected for us. And either one of those can create our backup documentation or reference material. So then the next step in our process is clearly defining our potential parameters. And that's our next slide, number 19, which is looking at what we're trying to control and what are the parameters that we need to set in order to achieve that control. So I've just kind of put together some examples here. That those could be time, pH, temperature, contact time, concentration, if it's, say, for instance, sanitation or a chemical kill, if it's product coverage, if we need to have a specific size dimension or availability of surface area. So say, for instance, if you're passing a product through a heat step and you've got dimensional control, either of the piece size or the bed depth or the bed width, we need to make sure we document all of those different parameters so that we know that we can recreate it or duplicate it on a regular basis and achieve what we achieved during our study. So as again, agency verification is going forward, they're going to be looking to make sure that we have, in fact, duplicated all the potential parameters. If, in fact, we're not going to conduct the study ourselves, that we feel our process is pretty routine, pretty similar standard to what goes on out in the industry, and that this has been done before, we're using pretty run-of-the-mill rote processes, equipment, and procedures, there is a possibility that you could have a minimal validation through implant data and studies, which we'll talk about later, but base most of your decision and program on documented criteria. That's slide 20, where we talk about what is the criteria for the documentation. So it needs to be widely accepted by relevant professionals. It needs to be the best available scientific data, meaning up to date, not created back in 1914. And is it reliable? So can we recreate the equipment, the products, the conditions that were used in the initial study? If it's so old that the equipment is outdated, or even, product dimension sizes aren't that commonplace in the industry any longer, or formulations have changed, then that's not going to be the type of documentation that we want to use as our reference material. Then comes the next slide, number 21, where we have to do a practical demonstration. So even if we're using documentation of scientific studies, at some point we still have to prove that that's relevant to our actual operations. We have to do some type of implant testing, observations, measurements, collecting test results, having those records available for review to show that, again, our plant conditions mirror those of the reference study, or we're creating our own reference data in order to justify or validate our process. So the next slide is number 22, showing a documentation example. So we're going to have a published scientific article stating that the time and temperature of a process and the level of pathogen reduction is our control. We'll have recorded documentation, which would be records confirming that the steam, say for instance, pasteurization process can be applied per the specification in the article in the plant setting. Now, microbial reduction can be used as a substitute for pathogen testing if there are, again, good scientific correlation studies out there showing that that is a reasonable substitute. Because if we're dealing with some exotic organisms that are not routinely, on our top 10 list, then we may have some correlation studies that says we don't have to go out and get these exotic strains. We can use kind of a simulation. But just be sure that that documentation, that study is clearly linked and out there saying you can substitute salmonella for, some exotic island bacteria that you may have encountered in imported ingredients. In the next section, we want to move on and talk about experimental design. So we've talked about how we need to mimic whatever went on in the control study or in our reference study. But if we're going to do that, we need to make sure that we've captured all the variables. And while we don't have to be as scientific, an expert on statistical process design and collecting every iteration and 3D control charts, we do have to be rigorous enough to capture the normal variation in our process. We have to collect enough data, which a lot of times just means time. So instead of thinking about, am I capturing this parameter to the upper and lower limit and every single iteration and combination of different factors, we know that our process is going to vary over time. And if we agree or decide that we will capture data long enough, we will definitely be able to show that we've satisfied that criteria. Whether that's 3 months, 6 months, we need to make sure that we're addressing any seasonal variation in ingredients. What would this be, environmental or temperature, seasonal variation in humidity and in, environmental conditions within our plant. In the wintertime, maybe it's very cold, and so it's a lot more difficult to achieve ambient temperatures and maintain. Or in the summertime, maybe it's more humid, and we may have more issues with this ambient microbiological flora, mold, et cetera, and mildew. So, we have to make sure that whatever those variations may be in our process, that we collect data long enough to capture those types of changes. And then that satisfies our experimental design. So our next slide deals with the collection of that data. And again, so the reason why collecting over time is better than even experimental design is because experimental design kind of involves forced control of variables. So you're going to undoubtedly put together scenarios which will never actually happen or exist in nature or in your process. That can obviously relate to non-representative data. So we want to avoid that anyway. So collecting data, again, over the natural variations in the process and the materials is actually a better way of achieving a robust data collection. Whatever the case might be, the most important thing is, again, that we're documenting all the variables so that we know that we've tested all the assumptions and that we have an understanding of what inputs are affecting our outcomes. Slide 25 is the initial validation, where we need to focus on, obviously, our critical control points, achieving our critical limits, and setting up our record-keeping procedures, corrective actions, and moving forward with our HACCP plan. So those are important aspects to validate as well, how we're going to, on an ongoing basis, collect data that we can review or verify. Some of our control measures will be critical control points, but as we move to the next slide, number 26, some of our control points will be within prerequisite programs. And what does this mean? We are all familiar with prerequisite programs, but sometimes we lose sight of the importance of them in that as we're doing our hazard analysis, we have identified that some items are not critical control points because we have an entire global program that is implemented by everyone on the food safety team or operations team, and that affects and touches and is involved in our product quality and safety. All of these inputs are important on a daily and everyday basis. The best example of all is always hand washing. And we call those prerequisite programs because we're not going to have a time delineated basis for people to wash their hands and record that, and that's not part of the process in the same way as heat from the oven. But it is a critical part of the process in that people are touching the product in certain industries or in certain processes all the time. So the condition of their hands is just as important as, say, an oven temperature or a chemical addition of preservative or some kill step or something that controls safety, or a sieve or a magnet. It's just as important, but it's not called a critical control point because we're controlling it on a more global scale and we can't actually measure it in the same way. So these programs or these activities are embedded within our prerequisite programs, such as our facility design. If we've got condensate and chip paint dropping from the ceiling, if we've got poor quality suppliers sending us high-level micro-contaminated ingredients, if we've got no specifications so we don't know what our incoming ingredients are, if we've got production equipment that is cast iron, lead paint, if we've got poor cleaning and sanitation, cross-contaminating our ingredient, and obviously the list goes on, we're so out of control, there's no point in establishing a critical control point because we have no point of control. So prerequisite programs are kind of like our baseline control. In this respect, moving to the next slide, it's as important that we document our prerequisite programs as it is that we document our critical control points. So having internal audits, keeping records of execution of our program, chemical, sanitation, concentration records, anything that we do on a daily basis that affects food safety needs to be documented so that it, again, can become part of that record set, that database that we use to verify that the program is working. So the next slide just gives us an example of a physical hazard validation. So say, for instance, foreign material control. The critical limit is 0. The control is removal via filters. That's going to be our CCP. And what we've got to do is validate that the size of the filter is capable of removing the size of the undesirable material. So again, that's validation. If it's a biological hazard, as noted on the next slide, number 29, our goal would be, say, for instance, E. coli removal in lettuce. Our critical limit would be to achieve negative colony count. Our control would be a chlorinated wash, which would be our CCP, and then our validation would be a challenge study to see that if we do wash with chlorine, that we can achieve a negative colony count. And finally, for chemical hazards such as an allergen, on slide 30, we've got, say, for instance, we have an allergen that we need to keep below a 20 ppm limit. Our control measure might be our allergen prerequisite program, which, as you know, can be pretty comprehensive in terms of how we segregate the allergenic material from the non-allergenic material, and labeling, receiving, storage, transition of production materials, I'm not sure if I said cleaning, but then finally packaging and, again, labeling for awareness. So that whole entire program is a PRP as opposed to a critical limit. Now, a critical control point. Now, we may escalate that to label review being a critical control point where we're actually identifying some final steps that we want to have in place. However, we may relegate it back to a PRP or an entire program or baseline program that's controlling a lot of different factors. on a more holistic level. And then how would we validate that? We would test products and contact surfaces to make sure that we're achieving that less than 20 parts per million exposure. So then on slide 31, we would transition into ongoing verification. So at this point, we have validated the program, and we've collected data over a period of time. And as we start to utilize this program that we've developed, this HACCP program, we need to think about ongoing verification, wherein the things that become important are the calibration of the instrument, observation of monitoring activities, corrective actions. All of these things are going into our assessment of whether or not we are implementing the program as we validated and designed it. So on slide 32, we're just reiterating that the parameters of the validation study have to be compared to what was in the reference study, but also we need to include things like raw material intervention steps from suppliers, validation of prerequisite programs, corporate studies that we validated at the plant level. So we need to make sure that we are still in keeping with our original assumptions as we move forward in implementing the program. Reassessment is something that is also very critical because we need to be sure that we haven't, along the way, implemented any changes that could invalidate our program, whether that be raw materials, formulations, processing methods, or even production volume. So running the lines faster, again, will reduce our time temperature correlation, and we may end up not achieving the results that we're expecting. So slide 34, when we're reassessing the HACCP plan, we need to also take into consideration that we maybe have new personnel, new packaging, new distribution systems, or even different consumers that we need to cater to. So all of these things can be an impact on our HACCP plan. And this is, again, folks required. Slide 35 references the CFR, which says that reassessment shall be performed by an individual trained in accordance with the 9th CFR standard 417.7, training Title IX, animals and animal products, blah, blah, blah. Bottom line is this is a documented part of the standard of the regulatory guidelines that we are required to implement. We've got to have, again, qualified, credible personnel doing the testing, and we have to meet the basic parameters of scientific validity. And we have to do this on a regular basis, i.e. reassessment. So most times that is annual, but anytime there's a change to the process, a reassessment needs to occur. So, for instance, slide 36 says that every establishment shall reassess the adequacy of the HACCP plan and hazard analysis yearly or when changes are made. Reassessment includes reviewing the scientific validation, documentation, and adding new information when possible. So as the industry is out there moving forward, we need to be moving forward with it. So questions we want to ask ourselves are, slide 37, does my HACCP system contain supporting documents for each CCP? Does it contain supporting documents that relate sufficiently to my product and my process? And does it identify critical operating parameters based on the supporting documents that I use as scientific support? The next slide continues with, does it contain critical operating parameters that are aligned with the reference supporting document? Meaning, have I documented, have I recorded, written down exactly what my equipment settings are, and can they be linked? Are they directly referenced in the supporting documents that I use? And finally, does my HACCP system contain critical operating parameters to support rather than contradict the selected critical operating parameters if I'm using multiple supporting references? Again, we're continuing to review our guidance, our FSIS guidance. Does my HACCP system contain implant demonstration data? So 90 calendar days is a good model. As I mentioned, 3 to 6 months. So at least 3 months, we want to be still in that validation data collecting mode. Does it contain HACCP system implant demonstration data for at least one product within each HACCP category? So again, we can't just validate one product. We need to validate each product that's significantly different from what else we're running. If it's different packaging or it has a slightly different, there's not a whole lot of things that you really want to overlook. If you've got size differences in your products or you have formulation differences, you really do want to consider those, but there could be some slight formulation like spices or seasoning. Well, even those, because different spices and seasonings have different microbiological levels that you might want to include in your HACCP validation. So there, again, every product difference is worthwhile in putting through your challenge study. I really wouldn't try to eliminate too many or consolidate too many. Does my HACCP system contain additional research data demonstrating the effectiveness of the process in instances where the critical operating parameters from the support were not followed? So for each HACCP category, identify at least one product from the category for which you'll collect implant demonstration data and complete a validation worksheet for such product containing the following information. And then there's some examples. So here we're talking again about making sure that we capture all the different variables, including our product variables. So what does that document kind of look like? We want to have the product, and we can also expand that to mean the HACCP plan or category. We have to specifically identify the hazard, specifically identify what point in the process that we expect that hazard to be controlled, and what are the critical operating parameters that impact that process, and then what is our scientific supporting documentation that has replicated that product, that hazard, that equipment or process, and the limits that we want to achieve. So slide 42, that was slide 41. On slide 42, we want to add into this equation the fact that, again, we've got some baseline important considerations, activities that we need to incorporate, meaning training and qualification of team members, calibration of instruments, an annual frequency, and review when changes occur. So these things have to be running in the background of our realization of our process. We need to consider the team that's validating the study and making sure that they're experienced, have the educational background, certification, training, whatever they need to do in order to do a credible job. And that slide 43 kind of captures those elements. Our instruments need to be calibrated, as shown on slide 44, annually prior to use, using a third party, implementing corrective actions when we find that our instruments are out of calibration, making sure that we disposition products when we find our instruments are out of calibration. We may not have achieved what we needed to achieve in terms of food safety, and that product may be at risk, and that we've used the appropriate methods, so we have followed the instructions as we needed to. On the validation itself, slide 45 indicates that it needs to happen. There needs to be a review at least annually or when changes occur. And always remember that we need to sign and date and have an up-to-date record revision history. So then slide 46, advancing to our summary. This is pretty much our wrap-up of HACCP validation and verification. And what we went over today was that we need to validate our program to be sure it works, to be sure that it will work. We need to verify it to be sure that it is working. We need to document it so that we can show that we've done these things. And mostly, we need to start with a good foundation. Slide 47 is your appendix of or knowledge assessment. You also have slide 48, which is an attendance register. So you can sign your name there and write down what the course was all about. Keep that with your training records to show that by attending this webinar, you have been trained on how to conduct validation and verification, and that you can train your team, implement these practices, and move forward. If you need to review or gain any more knowledge or just really delve into the concepts any further, there's a reference listing here that kind of talks through where you can get additional information and background on the subject matter. That goes on through slide 51, which defines the NACNCF, which was referenced during the presentation. And that's just the National Advisory Committee on Microbiological Criteria for Food. So they have been a tremendous resource in the creation of HACCP or the implementation of HACCP in the food chain. And so I wanted to be sure you were aware of who they are and what that means. So with that, I'd like to conclude and thank you all for your attention today, as well as for your patience with the delay. If you have any questions, I'm going to turn the presentation back over to Ron so that he can field those questions, and I'll be here waiting to hear what you need. Thanks again.
Thank you, Melinda, for the wonderful presentation, and I would also like to thank all participants for cooperating with us. Ladies and gentlemen, as Melinda informed, it's time for the Q&A to begin, and I request all who have questions for our presenter to click on the Raise Hand option, which is a palm-like icon at the bottom of your participants panel. Now, this is to ask your questions verbally, or you can also post in your questions in the chat column so that I can go ahead and read out the question to Melinda, and we'll get that answered for you. In the meantime, ladies and gentlemen, as we're waiting for the questions to come up, I sincerely request you to share your feedback in the feedback form that has appeared on your polling panel right now. If you can see, the feedback is just 8 questions, mostly multiple choice in nature, and it wouldn't take more than 2 minutes of your time to answer it. Now, this polling would remain open till the end of the session, so you can answer it even after the Q&A is over. We would be grateful if you could stay back a little longer and complete this feedback for us. Just a reminder to all the attendees, now if in case you do come up with any questions even after today's session, you can go ahead and send in your questions to me, and I will pass it on to Melinda, and we'll get that answered for you at the earliest. Ladies and gentlemen, if you do have any questions to our presenter, please go ahead and send in your questions to me through chat or click on the raise hand option, which is on your panelist column, and I will go ahead and unmute your lines. You can go ahead and ask your questions verbally to our presenter. Okay, Melinda, I don't see any questions that's coming up from our participants. Let's give it about another minute to see if there is any questions. If not, we'll go ahead and conclude today's session, okay?
Okay, that's fine.
Thank you so much, Melinda. And ladies and gentlemen, this is the upcoming webinars from Melinda Allen. If you do need more information about those webinars and what are the areas that are covered, you can visit our website, which is www.globalcompliancepanel.com. We have one scheduled for this Thursday, which is July the 10th. The duration is for 60 minutes. If you all are interested, request you to go ahead and visit our website and get yourself enrolled in this webinar. Okay, Melinda, I don't see any questions that's coming up from our participants. Before we could conclude today's session, do you have any final thoughts left?
No, I just want to say thank you again to all the participants, and I hope that the information was valuable, and that if they do have any questions, to please bring them forward, and that, as you said, I'm still happy to answer those at a later date if they want to provide them. And again, thank you.
Thank you, Melinda. Ladies and gentlemen, just another information. If any of you feel that your team members, colleagues, friends might benefit from this webinar, we are happy to inform you that it will be available in the recorded format. You can purchase it from our website, or you can call our toll-free number, which is listed on your screen right now. We welcome your suggestions and feedbacks on ideas on how we can improve our webinars. And ladies and gentlemen, just a quick reminder before you could log off today's session, request you to go ahead and share your feedback from the session. Your feedback is very important for us. On the note, ladies and gentlemen, we look forward to having you with us again sometime soon. On behalf of our presenter, Melinda Allen, and the Global Compliance Panel team, I'd like to thank you all for participating in this webinar, and I wish you all a pleasant day ahead. Thank you, ladies and gentlemen. Melinda, thank you so much for your time. You have a good day.
Thank you. Bye-bye.
Bye-bye.